A very good afternoon. The second lecture on genetic engineering of plants. In this particular lecture, we will be studying the plant transformation using the agrobacterium tumefaciens. And this is Dr. Nityananda Sastry, Associate Professor of Kelly College of Pharmacy, Belgavi. In the last class, we have discussed why plants are suitable for genetic engineering, what are the limitations of the conventional crossbreeding, what are the methods of the crossbreeding, what are the disadvantages, how these conventional crossbreeding are slow and uncertain, and issues that are related to the crossbreeding for improvisation of the plants and the traits in the plants, selection of the plants with the desired traits, and repeated backcrossing can lead to the problems. And we have also understood what is polyploidy and other conventional methods used in the plant improvement, and how the biotechnology promises a better opportunity to circumvent the limitations which are there in the conventional breeding. And also we have studied about the impact of biotechnology for developing new plant products using, and also the general concerns of the and issues of the GMOs. We all know, to re-emphasize what we have studied, we all have an understanding of what are the advantages of plant cells. Plant cells are totipotent cells, which means that each cell can can grow into a adult plant, and fertile transgenic plants can be produced from the genetic engineered cells. Therefore, whatever the traits which we can develop in these particular plants can be permanently incorporated, and they can be transformed into a genetically engineered plants. Desired plant traits can be passed on to the successive generations, and plants have a much better regenerative capacities than the animals. Therefore, they are more amenable for the genetic engineering, and there has been a long history of this plant breeding. Availability of wide variety of genetically modified plant strains are also indicate that it is a very feasible technique for plants. Plants produce large number of progeny. There are rare mutations and recombinations are found easily. So species boundaries and compatibility are not an issue with these particular plant species. And the advances in the molecular biotechnology, many more things can be done. Genetically determined traits can be introduced as a single gene or a cluster of genes. So to summarize, scientists have done, and there are so many varieties of newer plants, newer traits have been introduced into the plants, and we have many practical applications. To infer some of them, we have transferring the genes to the plants allows the innovations that are not possible by the conventional breeding. We have plants that produce their own pesticides. We have plants that are producing biopharmaceuticals. Today we have plenty bodies wherein the antibodies can be produced in the banana and other thing which can be used as a vaccines. So plants that produce vaccines, plants that produce strong fibers, for example in a jute there have been a lot of development of transgenic jute, and plants that produce the enhanced nutrition for nutraceuticals, they are of great advantage. And similarly we have plants that produce novel secondary metabolites. Many of the phytochemicals today are being produced which are of great market value through the genetic engineering technology. These are some of the suitable examples. So the points which we are going to discuss in this particular lecture will include how do we develop transgenic plants by using the plant transformation techniques, and what is the crown gall technique and how is it formed, what are TI plasmids. The transgenesis is carried out basically by a number of methods, but in this lecture we will be focusing on one bacterium mediated transgenesis, namely the agrobacterium tumefaciens, and the mechanism that is involved in the transferring the genes to plants. What are TI derived plasmids, which are the component of the agrobacterium tumefaciens plasmids which have been modified by the scientists so as to make them a better vectors for generating wide variety of vectors, and which can be used in the transgenesis generation of the transgenic plants. And we also will try to study how the reporter genes in these particular transformed cells can be made. So just as an overview, we will understand the plant transformation using the agrobacterium species. Agrobacterium species are a gram-negative soil bacterium, and they are usually plant pathogens. They are genetically transforms the plant cells. As we can see, quite often we come across these pictures, these type of plants, wherein there are crown galls, and scientists have found their roots have been uh, dominated by the existence of these particular species of the agrobacterium. So the transformation will lead to the crown galls, and they affect mostly the dicotyledons, the grape plants, roses, and stone fruit trees. So if we understand the mechanism of this crown gall and how this particular agrobacterium, which is associated with the roots of these particular plants, create the infection, scientists have mimicked or used them as a vehicle to transform the genes of our desired traits into the plants. And let us go into the mechanism of this particular crown gall formation. The initial step is attachment of the bacterium to the site of open wound. Simulation is done by the scientists in the laboratory, wherein the meristemic tissues or leaves are taken out and then cuts are induced, and they are subjected to this particular agrobacterium so that they can transfer the genes of interest. But in general, in nature, the initial step is attachment of the bacterium to the open wound, and it is believed that there are certain phenolic compounds like acetosingin and hydroxyl acetosinginosine are secreted by these susceptible plants, and they represent the products of the polypropanoid metabolism, one of the metabolism of secondary metabolite pathway for lignins and flavonoids. So they induce this induction of the viral genes of the TI plasmid. So we will see what is this TI plasmid. These are the plasmids which are present in this particular bacterium, agrobacterium tubifaciens. So we have the virulence genes that are located. If you look at into the structure of this particular genes, the particular plasmid, we see that there is what is called as a T region, and there is a virulence region, there is a opine catabolism, and there is the origin of replication. These are some of the essential features of a plasmid, wherein with the origin of replication is a must for the plasmid to replicate. Initial manipulations will be done in the bacterium, then the genes are introduced in the region of the T region, and for this particular T region we have what is called as a left T border and a right T border. In between these particular thing, we have auxin genes, we have oncogenes, we have cytokinin production, we have genes for opine synthesis, and together this becomes an octopine type TI plasmid. So they are essential. This particular virulent genes are essential for the transfer of and integration of the tDNA region into the genome of the plant cells. So these from the nature, the TI plasmids are isolated, modified to suit to the needs of the genetic transformation, and genes are introduced into the TI plasmid, transformed into the agrobacterium, and which are then subjected to the attachment to the plant of wherein we wish to have a desired modification. So the attachment of the agrobacterium tumefaciens attaches to the host plant cells, and virulence genes are introduced. tDNA is transferred. This particular part of the DNA is transferred by a process similar to that of a bacterial conjugation, and the genes which are transferred as a linear single strand molecule, and therein we have 5' end carries the right border sequence and the left border is at the 3' end. So just to have an overview of the pictorial form of this particular thing, as we can see, wounded plant cell produces a compound called as acetosyringosin, which will induce the agrobacterium attachment. there is a transfer of the tDNA, and then subsequently there is a synthesis of auxins, cytokines, opine, which will lead to the formation of tumor formation. Also, we can see that copy of tDNA is transferred and integrated at a random site into the plant chromosome. So the mechanism of crown gall formation has been studied and has been mimicked by the scientists to create newer varieties by introducing the genes of interest into the T plasmid. We have the formation of single stranded tDNA, which depends on the cutting of the strand at a specific sequences, borders which contain usually contain 25 base pairs, and its right border that is involved in the integration. So right border is involved in the integration of into the genome. The genes on the tDNA are activated after the genome integration and produces these genes that are responsible for the crown gall. So similar way, the unwanted genes are removed and the wanted genes are introduced into the T gene. Therefore, we can get the desired trait into the plants. So the genes of tDNA include some of the well-named known genes, TMS1 and IAM1. So these genes encode the enzymes that synthesize the auxins. The tDNA also contains the genes for opine synthesis. Opines are the condensation products of amino acids and keto acids. So we have the arginine and pyruvic acid. So octopine, arginine and alpha ketoglutaric acid, we call it as nopalin. Opines are synthesized within the crown gall and then secreted. They can act as a carbon and nitrogen source for the agrobacterium, because the bacterium which is there, it needs some of the sources. It is a mutual benefit for both the bacterium as well as the plant. The plant will utilize these particular materials as a carbon sources, and they can act as a carbon and nitrogen sources so that the organism will grow. The Ti plasmid has certain catabolism genes which are there outside the tDNA, and the unique mechanism make these particular plant cells for the production of unique carbon compounds which can be used by the agrobacterium species. So how do the Ti plasmids are used? How are we using this Ti plasmid for gene transfer? So if imagine there is an agrobacterium, each bacterium has got a genomic DNA and an extra chromosomal DNA. Ti plasmid are isolated. The plasmid is removed and cut open with the endonuclease, and the gene isolated of our trait, whichever is interested. Say suppose if you want to produce a beta carotene genes are cut and then introduced into the thing. A recombinant plasmid is being prepared and then retransformed into the agrobacterium tumefaciens cell, and this particular thing is subjected to interaction with the plant cell, so that the plant is put back into agrobacterium, and when mixed with the plant cells, you can create a callus culture or you can create a suspension culture. So the bacterium transfers the gene which we have introduced here into the recombinant DNA into the plant cell. So the plant divides and each daughter cells will receive the exact transformed gene. And pictorially, as we can see, it is an extension of recombinant DNA technology applied to the plant cells. And once we get callus cultures can be generated, further the plants can be plants are generated. So how to utilize the above unique mechanism for gene transfer in the plants? Insert a gene or DNA sequence of our interest, whether it can be a pest resistant gene like Bt cotton, Bacillus thuringiensis is a gene, is a toxin which is not digested by the insects, but however tolerated by the humans. So when these particular gene is introduced into the Ti plasmid and delivered into the plants produces a leaf which produces the Bt toxin, which cannot be digested by the insects. Insects will die, and therefore the crop damage is reduced, and that is how people will generate new transgenic plants. So good natural vectors are there, Ti plasmids, but however there are certain limitations to utilize the existing Ti plasmids, therefore they are modified. So phytohormones prevent the transformation to regenerate auxin and cytokine genes are to be removed from this TI plasmids. So whatever the genes which are not necessary for the transformation are removed, and genes coding for the opine synthesis are all not useful, as they divert the plant resources to the formation of opines. So both the genes are removed, and therefore the larger segments of DNA which are not essential for the cloning vector must be removed from the TI plasmid. So initially TI plasmids are isolated from the raw agrobacterium species. However, they are modified by the scientists to suit to the needs of an ideal transforming vector. And in addition, most of the manipulations have to be done at E. coli. Therefore, necessary mechanisms which are required for transformation and manipulation in E. coli are also introduced, and we have wide variety of commercial vectors which have been available, like for example Cambia vectors. These vectors can be used for transformation of genes into the. So just an overview of recombinant agrobacterium for the gene transfer. So you have the plasmid DNA, wherein you have the virulence gene, we have the high specificity region, and this particular thing, we have the tDNA. By the unique restriction thing, only a very good vector which is suitable for the transformation can be done. So there is a wound initiation will be done, and the recombinant bacteria, recombinant agrobacterium, which is being transformed with the TI plasmid containing the desired gene, will be subjected in the laboratory conditions, and there is a clone gene only present in the crown gall. So there is a you can generate the plants, and plants can be made into the cell suspensions. So after the inoculation with the recombinant agrobacterium tumefaciens, there is a transformation, and these transformed cells can be formed in the plates in the tissue culture media, so that once they can be regenerated, and this is how we generate a transformed plant cells. So basically the TI plasmid of the agrobacterium will be modified in a various way so as to bring about the transformation of the plants. The genes introduced into the plant genome are first cloned into the intermediate vectors for manipulation of DNA in E. coli, and that's the reason the recombinant vector, the Cambia vectors which are being chosen, are modified from the original vector so as to accommodate the mechanisms, free mechanisms of expression in the E. coli also. The intermediate vector is then transferred into the agrobacterium, which is the main which has the virulence genes which will be useful in transfer gene. So in as a resulting there are co-integrate and many derived vectors are also being done. So essentially these particular vectors, co-integrative vectors, are generated, and which are more in the subsequent classes we will understand more in depth about these particular co-integrative plasmids. But essentially agrobacterium is a gram-negative bacteria which has got certain plasmids, and these plasmids are isolated, modified, and to suit the needs of the genetic transformation, which will also incorporate the necessary manipulations in E. coli, as well as the introduction of the genes, and unwanted genes are removed. As a result, we will get a transgenic plants. In conclusion, plant biotechnology will not only have an enormous impact on traditional plant breeding, but it will also be used to create newer plants. We have opportunity to create the newer plants by introducing newer traits, and agrobacterium mediated gene transfer is one of the most popular technique for generating the transgenic plants, and we can use those plants for bioprospecting. A variety of beneficial transgenic cereal crops are there. We have transgenic commercial and medicinal plants using different techniques have been developed, and therefore it is essential for us. There are possible innovations, patents are possible. You can have the money, knowledge based products can be generated through this plant transgenesis. Therefore there is a need for us to understand the mechanism of crown gall and also the uh, techniques that are involved in generation of the plants. Field has tremendous scope and opportunity. So I advise you all to go more in details with the molecular biology, which has influence and an understanding of the plasmids, more so with the TI plasmids and RI plasmids, which are associated with the bacteria. And there may be other bacteria which are equivalent to that of agrobacterium. Even they can be explored. So at every stage of these particular steps, we do have an opportunity to understand the transgenic plants. Plants can be generated genetically by introduced genes into the plant cells that regenerate into a fertile plant. Variety of techniques are available to introduce the genes into the plants and have them expressed in the genes. So we have most notable is we all know that whole Southeast Asian countries are suffering from night blindness, wherein there is a precursor of beta carotene can be converted into the vitamin A, which is required for the vision. So in order to people have developed what is called as a golden rice using these particular techniques. So where variety methods are there, but however agrobacterium tumefaciens is a natural genetic engineer of plants. So people have exploited. We have wild variety of these particular agrobacterium tumefaciens bacterial genes, and the genes can be modified for the delivery of the genes that make plants insect resistant, so that we can reduce the crop damage. Viral resistance, we have examples of GM papayas and other thing. We have GM potatoes, GM brinjals and other important herbicide resistant plants with improved productivity, and products with plants with novel metabolites, plants which have been improved strength of the fibers, commercial plants. So many plants have been generated using the genetic engineering. Thank you for your patient listening.